Good evening to all the faculty members, organizing committee members, Dr. Ravi Mohan, who is the organizer and controller of the whole session, and the foreign faculty. SOGO has been there for almost 2 years now, and we have been arranging various academic meetings. Of course, for this last but one meeting for the year 2021, we thought we should discuss on genetics and urologic oncology for that matter. Having said that, the whole program was designed, and Dr. Ravi Mohan was interested to bring out the practical points to the ordinary urologists, uro-oncologists, and clinical practitioners, so that they have a clarity of thoughts, because so much amount of information is flooded continuously, and by the day it's increasing, and one really at lost where to pick the loose threads. And I must compliment Dr. Ravi Mohan for doing the excellent job, and he's put these all threads together, and we're looking forward to this exciting discussion, and there will be many takeaways for us, and we can capitalize on that. So with this remark, I welcome you all and request Dr. Ravi Mohan to take on further. Thank you very much.
Thank you, Kulkarni sir. And Dr. Raghunath and SOGO team for giving this opportunity. I think it is well known that we're all constantly chasing the goal of precision medicine, and as we're doing that, and with the better understanding of genome and the various genome analysis techniques, it has enabled us to better understand bladder cancer, prostate cancer, and almost all the genitourinary cancers. So with that, the theme of this meeting was to enable our fellow urologists with at least some take home that where and how and when should they do the genetic testing and how it is applied in clinics. To do justice to this topic, we have eminent two international faculties and 2 national faculties. Professor Ash Tiwari, I think the audience won't require any introduction for him. He's a well-known figure in India. But I have the honor of introducing him. He's a chairman of urology at ICA School of Medicine in Mount Sinai, New York, UK. And we all know that he's got a specific interest in prostate, nerve-sparing prostatectomies, and various techniques have developed. And he's a very avid, his present work is on genetics in prostate cancer. So he, unfortunately, he could not make it in person, but we have 2 or 3 of his fellows, as well as his associate professor, Dr. Sujit Nair, who would be there in the meeting. And Professor Ash has shared his meeting, recorded meeting version, so we'll hear him through. Then we have Professor Dan Theodoscu. He is the director of Samuel Austin Comprehensive Cancer Institute and overall leader of CDARS. I am sorry that I'm not able to spell it correct. CDARS Simoy Cancer Enterprise in California, which is the top 10 rank amongst the top 10 ranked in the cancer program in the United States. And we are honored to have him. Thank you, Professor Dan, for this. He will be speaking on cancer genetics as applied to bladder cancer, and he has selected a specific topic for that. And then we have Professor Colonel Anurag Mehta. He's the director of laboratory services and molecular diagnostics and transfusion services at Rajiv Gandhi Cancer Hospital in New Delhi, India. He would be talking on cancer genetics with respect to kidney cancer. Following which, we would have a panel discussion of 2 or 3 cases as per the time permits. This will be done by Dr. Sanjay Adala. We all know him. Sanjay is a very efficient manager of his whatever task we give him. He's presently the director of, he's a uro-oncologist and a robotic surgeon at Apollo Cancer Institute, Jubilee Hills, Hyderabad. With that, we have from SOGO team, Dr. Ginil and Jagdish sir. So with that, I would start Professor Ash Tiwari's presentation, and then we can have question and answers in the chat box as we go along. Thank you.
Sorry, is there a problem with audio?
No audio.
By the knife.
No audio.
And in my case, with the robot. With that
Is it heard now, sir?
I started more in depth about what is happening in the genomic level, and I will give you summary of the journey which I have taken. What we need to understand that I'm in front of you, and I am some total of about 30 plus trillion cells. My body is made up of at least 30 trillion cells, and many of these cells every minute are dying. Not only they are dying, they are being recycled back. So this process of my cells staying in a constant lively stage, some of them dying and some of them are coming back. This entire process is a very complicated process, and what you are seeing on the screen on the right side is a cell which is a very complicated structure in itself, and as it evolves into from one cell to this 30 plus trillion cells, it differentiates into different kinds of bodily organs. Some of the cells become the blood cells. Some of the cells become the eye cells, some kidney, some skin, some liver, and some prostate. These cells, they started from an original one sperm and ovum, evolved into a trillion cell factory in which there is a social system which exists, in which one cell chooses to float around in the body carrying oxygen. The other cell works on excreting waste products from the kidney, and there's another cell which is a metabolic factory in the body and makes the liver. And then there is the skin, there are body's defense mechanisms, there is a nervous system, there are bones, there are accessory organs. All that has evolved due to a very controlled differentiation process which our body, our species has learned over millions of years. But this process is not perfect, meaning whenever there's this new cycling of the cells which is going on, they generate back into its normal form, but sometimes there are mistakes which happen. And those mistakes can actually result in cancer. And that's the reason, as a surgeon, as a scientist, I have to learn about what is happening at the basic molecular level, which can impact the whole bodily function. And the other side of the screen is showing something what we call cell cycle process. Most of the cells, they are staying dormant, meaning they are doing their job, but they have no desire to multiply. But there are certain cells in the skin and certain cells in the lining of our gut, the certain cells in the bone marrow. They have to multiply again back because they are having a issue with the cell death, and the cell regeneration needs to happen. And this entire process of a resting stage versus what we call a G1 and a G2 stages, these are the cell cycles which is very delicately controlled. Not every cell can go on to a synthetic phase or go on to a G1 to the G2 phase or go on to the mitotic phase. There are certain checkpoints which exist. And these checkpoints are at 3 different levels, and these are some kinds of proteins which actually control the entire process. It checks on whether there isn't enough organelles present before the cell is ready to divide. There is enough DNA made and duplicated before the cell is ready to divide. Has the DNA duplication, has that process been taken through the quality checks or not? Because we don't want to create a cell which is not the perfect form. That entire built-in check and balance mechanism is what we call our life. And understanding about that life is the journey I'm taking you through the cancer genetics discussion. If I have to sum up, why are we having this discussion as a surgeon, as a scientist? It can give us an insight as to why prostate cancer happens. Genomics will help us in understanding who is at risk, how we can better diagnose prostate cancer. We have a diagnosis of prostate cancer and we can use genomic tools to make a better prediction as to which amongst these 10, 20 patients is likely to be a very advanced growing cancer versus one which will be very indolent. Genomics tell us we can manage the recurrence better just because we know what kind of a cancer has come back. And today, we are able to develop certain treatments which are targeted towards a particular genomic subtype. And as the end of this discussion, you will see we can even devise body's own defense mechanism to fight against prostate cancer if we understand the genetic detail what we call first-line genomic vaccines can be made. So genomics is not just an intellectual exercise. It's going to help us be a better doctor, better surgeon, and a better clinician. Let's go through this journey. I'm showing you on the left side what I get to see as a surgeon. As a surgeon, I see a structure, I see an organ, I see facial planes, I see tissue, where one structure ends and the other structure starts. On the right side, what you are seeing is a gross specimen which has come out, and that is the gross pathology, which is telling me that I have taken the organ out, and part of that organ is a disease, what we call cancerous in this case. On the lower left side, what you're seeing is a zoomed-in mechanism. First I saw the structure, now I'm seeing a gross image, and now I'm getting deep inside under a microscope. By magnification, I can see structures that where the cancer is, how cancer is trying to escape, and if cancer has become from an incurable to a curable to an incurable cancer because it has broken through the anatomical boundaries. On the right side, what I'm trying to suggest that all that is happening in an organ, in a tissue, in a microscopy actually is driven deep down inside the cells by certain proteins. Mind it, those proteins are making a cell become from a normal to a rogue cell. And that understanding of those proteins, those pathways, those molecular pathways is where the genomics comes in. So with this background, I want to show you a first ever case of a prostate cancer which was diagnosed in about 2,700 years ago. And that happened to be a king of an area which was between the Russia and Afghanistan. And that was known as the king of Azran. That king was a very strong person, but was the first case of documented prostate cancer in which we have been able to confirm that because of no other method but for the genomics that allowed us to confirm that he had a prostate cancer. What you are seeing in the bottom is the journey which happened in that patient from a single cell to a clump of cells to a more invasive pathway, and cancer became totally out of control. And on the right upper side, what you're seeing are the bones which became brittle, eaten away by the cancer, and that finally killed that king. Strong person, prostate cancer, started in a cell, started in an organ, grew beyond the organ, and overtook the whole body. That all can happen, and I want you to take through this journey of going inside the human body. What you're seeing now is the bladder, then you are seeing a prostate, and in the prostate, what you are seeing is one rogue cell decides to become 2, 2 becomes 4, 4 becomes 16, and then they grow to become millions and sometimes billion cells. Up to this point, it's all okay, but then it gets to a point where it reaches the blood vessel, reaches a nerve, and gets out of the boundaries of the organ where it actually took the origin from. Now it is becoming a problematic cancer. It is becoming what we call an invasive cancer, and then it will become a regional cancer, and then finally it will become a metastatic cancer, which will go to any part of the body. It went to the bones just like it went in that King of the Azra. How that single cell evolved from a very normal prostate cell into a rogue cancer cell became so angry that it took over, broke through all the control mechanisms, and then went on to overrule the whole body, and in that process killing the human. That all was happening because of certain changes which were happening inside the cells. And as a scientist, we can understand those changes, and that is what we call the genomic details of the cancer biology, which can have an impact on the final outcome, because there are certain treatments which may be there if we understand what exactly is happening inside the gene, which is abnormal and can be fixed. That's the reason we need to understand the genomics, and I want to bring you back into the discussions about the proteins. Everything which happens, you are watching this video, and this video is being watched by your eyes, and there is a protein in the retina known as rhodopsin, and that is changing the chemical structure and creating light into your neurons. It's going inside through your nerves, inside your brain, and the neurotransmitters are acting on it. The very fact that I can speak, I can move my hand, it's because of certain muscle proteins are acting and allowing my voice to come out, my fingers to move around, and I can change my gesture to look at it. All that is happening because of proteins inside the cells. Those 30 plus trillion cells I talked about, they have proteins inside. And the proteins are very, very important part of our body, because not only that, they can allow us to see or hear, but they make the structure of the cell. They act as a messenger of the cell. They are involved as an enzymes. They are involved in the cell communications. One cell talking to the other cell. Hey, oxygen has arrived, or an invader is there. All that messaging system is happening through the enzymes or through the growth factors or through the signals. They're all made up of different forms of proteins. Those proteins can also be very helpful for us to build up an immune mechanism so that it can fight an invading bacteria or invading COVID virus. All that is being managed and orchestrated by proteins. So proteins are our acting chemicals. Guess how these proteins are made? That is where the gene discussion comes in, because proteins are not only involved in the day-to-day functioning. As I said, there are certain checkpoint inhibitors. The process of entire cellular division itself is controlled by the proteins, either as a structure or as a messenger. And this entire process is controlled or coded by the genes. And that's what we need to understand, that each one of us have two books of codes given to us, one by my father and one by my mother. These two books have the entire genetic code written to us so that we can read it and we can copy it, and then we can create a specific protein whenever and wherever it is needed. Each one of these books has 23 chapters, 23 chromosomes. These 23 chromosomes are coming from father's side and from the mother's side, but these chapters have got small paragraphs. these paragraphs focus on one particular recipe. Let's look at the recipe is we want to fight a virus. We have a different kind of chemicals we need to produce. We have little lack of oxygen because we have gone to a higher altitude and we need to get in more RBCs in our body. A different other kind of protein needs to be produced. This all recipe in a very perfect form is written in our genetic book. This is our genome. And the alphabets in which these paragraphs and chapters are written is known as language of nature, language of life, or what we call a DNA. Each one of these chromosomes is a very complexly folded form of an DNA that is wrapped around, and I'll come on to as to how this wrapping even can have an impact on how things are functioning. But this DNA has segments. These segments are what we call genes. A particular paragraph of a particular chapter talks about a specific functioning, and these are what we call genes. And guess how many genes we have? We may have more than 20,000 genes, but at least these 20,000 we know of. And these 20,000 genes are coded or written by the DNA which we have. And the intermediary of this DNA is the book. We have a scanner or a reader, which is an RNA, 3 different kinds. They get the job done inside the nucleus, get the RNA out, and go and produce protein, our favorite protein. And that entire process is very complicated, and because DNA message through RNA is making a protein, but there are only 20 different kinds of amino acids. And these amino acids can be recycled, repurposed, refolded, recombined, and then shapes change to 20,000 of the proteins. All that is happening inside those small cells of ours. And this entire process is a controlled process. So that's the reason we wanted to understand how this process is being controlled. Book exists, but we don't take care of the book. The DNA messaging gets a little bit distorted. Message was there, but the reading didn't happen very well. So what we call a mutation happened. A particular segment of a gene was either misread or over amplified, or chromosome because of some reason got shattered and then got recombined, and the net result was there was a distortion in the actual true meaning of that gene, and that's exactly what we call mutation. Mutations happen every time. Mutations, they happen in our body all the time, but many of those mutations are not meaningful. Some of the mutations are meaningful, what we call the driver mutations. And some of the mutations are not compatible with the life. So what's happening? While mutations are happening, there are different kinds of mutations can happen. You read it wrong. When you typed it wrong. It's like in jumbled up typewriter in which the two alphabets are writing a wrong message. So this is a single alphabet is being transcribed wrong. But if a small segment is the problem, that entire thing will produce a protein, but instead of producing a squash, it will add sugar to salt. And guess what? That drink is not going to be very useful because it was supposed to be sweet, but somewhere the letters got mixed up. And if that happens in a very relevant gene, which is producing a protein which is critical with the life of that cell, we cannot let that cell go on. And that is where the control mechanisms, they come. Body has a very good control mechanism. Not only it's doing a proofreading more than once, it is also looking at the final product. And if the final product has a problem, it has to make a decision. Is this cell fixable? we have a mechanism known as the DNA repair pathways? Or is this cell not compatible? It's so defective that if we continue it living, it will create problems. Then that cell has to die, and we steer that cell towards what we call apoptosis. That cell decides, you know what, I live my life, I have a defect, and if I don't commit suicide, I will be damaging my neighbors. So this is a social system built in our millions of years of evolution, and this entire process is very well controlled. So what we learned, proteins were important, genes are in the chromosomes through the DNA, mutations can happen, but body has a very decent safeguard mechanism of dealing with these mutations. So I started getting more and more involved in this gene and how things are happening. So genes are the one which are controlling the proteins, and one cell to the trillions of cells, there is a total art in how this entire DNA is packaged. By the way, I mean, it's a more of a trivia question, and basically each one of us have enough DNA folded very meticulously inside our cells that if we unfold it, we can go from here to sun 300 times. Enough length of a DNA exists in each one of us that if we unfold it, that distance will be from here to sun, not just one time, 300 times. So basically, there is a lot of information is kind of arranged and coded within our. Every now and then, DNA special protein around it, what we call histones. These histones, they allow you to kind of do the packaging in a way so that it is compressed within a very small segment. And sometimes the proteins around the DNA also make sure that not everything which is out there in the DNA is coded. So this is a very precise machinery, and we as a surgeon scientist, and now you all have to comprehend it. So I'm trying to simplify it as much as I can so that you get the bigger picture. Obviously, there are textbooks and books for us to get into the deeper dive into this. So DNA is an important part. Nucleosomes, histones are very important part of this entire factory. There is a lot of DNA in our body. There is a lot of methods in which this entire thing is controlled. I also want you to know, without getting into too much of a detail, it's like very early on in our career, we were taught how to read the EKGs, and we were given the stethoscopes to hear the heart murmurs and the sounds and all the signs and symptoms of the entire general exam. Same way, a new field has come up, and there are different ways of looking at the gene. There is a phenomenon known as a whole genome sequencing. There is an exome sequencing. There are even what you call a methylation for the histone sequencing. And there are ways to get into the single cell and what we call a single cell sequencing. So we know that the DNA has genes. Genes control protein. Protein is important for this timely and regulated duplication of the cell, which is the reality of life. Every body has cells which are dying, and some cells need to come up. In that process, certain cells misbehave, and when they misbehave, they become cancerous. And my next transition point is, what do we mean when things become cancerous? And I want to simplify it. There is a very important paper which has been written about the cancer, and cancer cells share certain basic tenets why they become cancerous, and these are known as hallmarks of cancer. There is a very complicated diagram on the left side in which we talk about that there is a phenomenon of overcoming the growth suppressors. There is a problem with the immune evasion. But I will simplify this entire process into imagine you are having a car ride. You are driving, and at a curve, you're supposed to take your foot off the pedal so that the accelerator slows down. Suddenly you realize that actually the car's accelerator is stuck. If that happens, car will not slow down and will keep going. Same thing is happening in a cancer cell. Somehow cancer cells were supposed to slow down in a division process because there were enough number of cells, and certain accelerators are stuck. And we call them oncogenes. Oncogenes are certain proteins, certain genes, certain molecular pathways within the cell which just keep telling the cell divide, 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 divide. That should have slowed down, but something has happened to that cell that it is not slowing down. Okay, you cannot take the accelerator off, but you can, in that situation, apply a brake. And you apply a brake, and then suddenly you realize that the brakes are also not working. What I mean, that in a normal cell, when there is a hyper-accelerated pathway which is active, there are certain brakes which are built in, and these brakes are known as tumor suppressors. Tumor suppressors can block the entire pathway and then say, hey, you know what, you need to slow down. But cancer cells somehow figure out how to even override that control, and what is happening, that these cells, they don't stop. So on one side, there is an oncogene which is driving the mutations, driving the cell growth. And the other side, the normal control mechanism is also not working. What we call the tumor suppressors are not working. I'll give an example of an oncogene. Is a RAS is an oncogene, PI3 kinase pathway is another one which is involved. The P10, a P53 are what we call irregular tumor suppressors. Then comes the third thing, that okay, sooner or later, if you keep dividing, the normal cells have a limit known as limit of about 60 to 65 cell division, after which cell runs out of its dividing ability, and then that is controlled by something known as telomeres. Telomeres at the end of the chromosome, and if you don't take care of it, it frays and the DNA itself cannot divide, and it's known as the Hayflick's limit. That limit is there because of shortening of the telomeres, and we hope that the cancer cell after X amount of divisions will run out of the telomere and it will stop dividing. Now, cancer cell has figured out how to rebuild the telomeres. So the telomerase enzyme is there, and that enzymes keep regenerating back that portion. So multiple things are going on, and then you hope that it will run out of the gas. The carburetors are problematic and it will run out of the gas. So what we call the normal energetics of a cell is that the mitochondria works under aerobic conditions, and the aerobic conditions produces certain ATP, and cell needs those ATPs in a very efficient manner. Cancer cells have hacked the carburetor, and they can work in a non-aerobic pathways, and they produce more lactate and all those things, and that lactate is used for some other kind of a structural phenomena, and they have rigged the whole carburetor system. So this entire process is happening inside a cancer cell, and we need to understand what's happening and what is driving at the genetic level. Then think about, you are driving this car and it is just going fast and fast and fast through the road, not respecting any kind of a control mechanism. You hope sooner or later the traffic police will get to you and will finally force you or somehow manage to stop it. Yeah, that's supposed to happen to the normal cell, but the cancer cells have figured out how to not be seen by the traffic cops. So our immune cells are supposed to stop it, but the tumor cells have figured out how to hide their antigens so that they are not exposed. And they can go on for a long time. Guess what? This all is happening in a cancer cell which has just refuse to follow the social rules of not dividing beyond a certain number. Next thing comes that the normal cells will run out of gas, meaning they need oxygen, and after a while, when the number of cells has grown beyond a certain limit, it will be needing more oxygen, but there is no more oxygen to come. But that's where the cancer cells become a little tricky and conniving. They will produce what they call tumor angiogenic factors. So they will create chemicals which will create sprouting of the capillaries so that they get a preferential blood supply of more oxygen coming to them, and that phenomena is known as angiogenesis. That all is happening because of certain enzymes, certain proteins, certain genes are acting and helping cancer cell in getting all this achieved. Few other things are going on. One of them is that the gene itself in that cancer cell is not very stable, and we call it genomic instability happening. Other thing is, when there's so much of an unregulated cancer growth is happening, the body produces some inflammation, and inflammation now colludes with the cancer and making it even more aggressive. So pro-cancerous inflammatory process is one of the hallmarks of the cancer. But to me, one of the biggest tools which cancer cells they use to become from an one solitary cell to a billion cell mass is what we call this vehicle, which was only trained to drive on a highway, on a road, changes from a regular cell to what we call an all-terrain vehicle. What is happening? The cell which started early on a de-differentiated cell has become differentiated to be a prostatic cell, which is supposed to be not move around. It's supposed to sit on a basement membrane, produce certain chemicals and assays and all those things. This cell is regressing backwards to a point that this regression will result in what we call an epithelial to mesenchymal transformation. This car is now becoming an all-terrain vehicle. It can grow within the collagen network outside into the tumor microenvironment. It can drill holes using the inflammatory cells and other methods and metalloporphyrins to go outside the capsule. It will collude with the nerves to find its way out. It will then go to the lymph node, it will go to the blood vessel, and now it has become a metastatic niche. It's circulating everywhere, and finally, sooner or later, it will find a new home, will produce this entire process one more time, and this is what is the journey of the cancer. And this complicated slide in front of you summarizes that many different proteins, many different genes, many different pathways are what are doing this entire damage to the human body. Okay, it was a little complicated, but we had to go through this because in order for me to explain, I myself went through this entire process, and it all started when I started collecting tissue during my robotic surgery to understand what is happening deep inside the cell. I started taking tissue from the different parts of the prostate. I started doing a single cell genomics, and I started looking at the what we call the copy number variations, and even in one different location, one prostate, one cancer, different parts of the cancer were different clones. That resulted me in understanding that this is in clonal evolution which is happening. That brings me to a different thought, what I'm going to introduce, that prostate cancer carries name prostate cancer in 10 different patients. You think they may have a different stage, they may have a different Gleason grade, but at a fundamental level, at a molecule level, at a gene level, at a protein level, at a pathway level, these 10 cancers will be very, very different. And I'll get to that, why it is important. But cancer may become a very aggressive cancer, taking a totally different pathway. And that is what is being shown here. It's one of the first papers which I wrote with my colleagues in Cornell and at Broad Institute in Harvard system. These are all my patients. They have a different Gleason grade, but when you look inside, what you are seeing here is known as sarcograph. Circogram is an EKG for the genes. Each one of those numbers represent one chromosome, and it is also showing whether a part of it is deleted, added, translocated, and if it has translocated, where it has gone and got attached. This is what the circogram is, and just looking at it grossly, look at the right lower one and the left upper one. There are two different kinds of circograms, and that's exactly what is happening at the genomic level. 7 different patients, 7 different genomic types. So this phenomena is known as an inter-tumor heterogeneity, meaning between these different patients, don't expect them to have the same kind of a cancer. And we started looking at what exactly was going on, and these are some of the papers we wrote as to what is happening at the mutation level and what was happening in the epigenetic level, and how exactly this entire process was happening. One interesting thing came out, what we call it punctuated evolution. Certain mishaps were happening early on, then small mishaps were happening a few years later, and then finally one more disruptive process happening and the whole chromosome got shattered. And once it got shattered, it tried to refix it back, but when it was fixing it back, it was connecting ear to the nose and finger to the thumb, and that kind of a restructuring is known as chromothripsy and chromoplexy. And net result was a very abnormal DNA was being produced, and that contributed to the genesis of a more aggressive prostate cancer. And throwing into this the discussion about the androgens. Androgens in prostate cancer can impact almost all of these pathways which I have talked about. The oncogenes, the suppressors, the inflammation, the angiogenesis, genomic alteration, all that can be mediated or propagated by the androgens, and that happens to be the main treatment modality what we are dealing with in prostate cancer. So this just tells you how intricately involved this entire metabolic pathway is. The only reason I can talk about it is because of understanding a little bit more about the genes. I don't want to re-go into this entire process, but I will show that growth factors of cells becoming 1 to billion is happening on the left side, but what I worry about what is happening on the right lower side, how the cancer cells are becoming from an average cell, which is number increase in mass, to a more invasive metastatic cancer. And this all can be quantified by different genetic tool which we have now. So that's the important part. And we finally know that in a simplified version, there are a few different kinds of prostate cancers. I'm not talking about the Gleason grade. I'm not talking about the stage of the cancer. I'm talking about that 71% of the cancers will have a bottom line abnormality in the androgen receptor molecules. But 49% of the patients will have something totally unrelated, which we call the PI3 kinase pathway. Just to connect you back on what is this PI3 kinase pathway, is insulin receptors, sugar, insulin activates a metabolic pathway inside the cell, and that is known as the PI3 kinase, AKT, mTOR pathway, and ultimately it manages the certain parts of the metabolic system. That I will call it sugar addiction. So 49% of the prostate cancers have something abnormal in the PI3 kinase pathway, which is something to do with the insulin pathway. Then 13% of the patients have an abnormality of what we call a DNA repair pathway. You remember early on I was saying that the DNA is checked for the proofreading, and if it has a minor abnormalities, there's spelling mistakes, it can be corrected, and that entire process is known as a DNA repair pathway. Those DNA repair pathways are also known as the BRCA, because that is very famous group of certain genes which are involved in the breast cancer discussion. But the similar gene group is involved in prostate cancer also, and 13% of the patients with prostate cancer who have an aggressive prostate cancer actually have a problem with their fixing mechanism of DNA repair. So DNA is being damaged in a normal form, but its repair mechanism is not working, and then a mismanaged damaged DNA is allowed to become a living cell in the community. And that sometimes happen to carry a certain particular genomic abnormalities of the one which we just talked about and becomes a cancer. Then we talked about the cell cycle discussion. You remember that there are 3 different cell cycle blocks so that the cells are proofread and allowed to go to the next level only when they are good. There may be a problem in those pathways. And if that is so, 21% of the time when we are finding that there is a problem there. How does it matter? DNA repair pathway, there are certain medications which are out there. DNA repair pathway also leads to what we call a mismatch issues, or cells become a little bit more recognizable to the immune system. So mutational pathway goes up. So DNA repair pathway, if we recognize has an issue, we have certain medications built in for them. Someone has a PI3 kinase issue and we are just depriving them of 71% of the androgen receptor. They're not going to budge. We need to understand what is happening in a molecular level to a particular patient so that we can give a more targeted treatment. Even in the androgen, there are issues with the splice variants. There are so many details which we are getting that we can tailor the treatment not for a organ, not for a tissue, but for a gene, but for a chemical, but for a target. And that is where this entire field is evolving. And this is just a good example of how understanding of the genes allows us to reclassify patients. Then comes a discussion about the genes, and there are 2 words I want you to understand. So far, all what I have talked about is what is known as a somatic mutation. The genes within the cancer are abnormal, rest of the body is doing fine, and those abnormal cancer genes are somatic mutations, and they took over the process and they are ruling right now. But there are certain mutations which are present right from the birth, and these are known as the germline mutations, meaning they are present in almost every cell in the body. Retinoblastoma gene you have heard about. There are Lynch syndromes in which there are certain abnormalities built in, inherited by us, which predispose us for more cancer. So that is known as a germline screening versus going deep inside the cancer, which is already developing, and that's known as a somatic screening. It's very important for us to understand both are genomics. One is in all the cells, which is a germline. Another is in the somatic cells, which is happening within the cancer cells. So what are the practical utilities of this entire discussion? And I will take a couple of scenarios. I'm not taking all of the scenarios. Many times we do the prostate biopsy. Biopsy comes back to be negative, but the patient's PSA continues to rise and we don't know how to advise them. So prior negative biopsy, we have a tool which is based on what we call an epigenetic or a halo effect of the DNA methylation project, in which tissue which is not showing cancer on a microscopic level on a genomic level is still showing certain changes which are not yet there for a pathologist to pick up that is a cancer, but it is showing a predisposition that in next 3 years, 5 years, this patient's tissue is likely to change into what you call cancer. If that is positive, you will have a different kind of a follow-up regimen into a patient, and at least in United States, that's available as a confirmed MDX. And it's a good tool for me to use whenever we are dealing with a patient with a prior negative biopsy. We found a cancer. Cancer is a Gleason 6 or a Gleason 7, 3+4. What to do with that cancer? There are many different kinds of treatments which are available, and the patient could be a very adequate treatment for what we call a watchful waiting or an active surveillance. Some of them would like to have focal therapy. Some of them will be a very good candidate for a nerve-sparing robotic prostatectomy, and some of them needs irradiation. how to kind of quantify, substratify a patient who already had the diagnosis of cancer. And that is where different other kinds of tests which have become available. And one of them is known as a Polaris. And there are more tests. One of them is known as an Oncotype DX, which has different genes which are associated with the worst outcome, and there are certain genes which are associated with the good outcome. Stromal response genes and the proliferation genes are associated with the worst outcome. Androgen signaling and cell organization genes are associated with the better outcome. So knowing what kind of genes which are there in that Gleason, same, similar looking, but very different inside, can have an impact, and we tend to use a lot more for our selection of the patients on our active surveillance. The third one is a Decipher. It has multiple genes and it has 22 different kind of an RNA biomarkers which it has used. It kind of gives a risk profile as to what's the likelihood of this cancer will behave more aggressively versus less aggressively. And this can also be used not just in the biopsy tissue, but we tend to use it after a radical prostatectomy, understanding what's the likelihood that this patient's cancer try to come back. This is all happening because of our understanding of the gene, and it gives a big picture. Polaris, OncoDX, Decipher, all of that combined or individually will help us in choosing the patients better for active surveillance. It will help us in identifying which patient may actually need what you call an adjuvant treatment. Which patient can need a neoadjuvant treatment. There are newer kind of treatments which are becoming available so that the recurrence rates are lower, and that all can be tailored through this approach. Even looking at the pathology, there are certain pathways like Ki67 and the androgen receptor. You can go deep dive into an image analysis of the pathology and upgrade that with the what you call an immunohistochemistry. And you can get to the genetic level of that. And your pathology becomes an augmented pathology. It becomes a genomic guided pathology, path genomics. And I have been involved in this paper with my colleague Carlos Cardon, who is the chair of pathology here. It makes an impact in us making a decision just looking at the histopathology. So I won't just say this is a Gleason 6. We have a little bit more angle into that. And then comes a new field which is evolving, and I'm very happy that my group is quite involved. The MRI is becoming more and more sophisticated, and looking at the MRI images, looking at the biopsy images, we can combine all that and come up with what you call a better understanding how it correlates with the genomics. So we did all that. We correlated the genomics, PIRAD, and histopathology, and found that there are different pathways which are being involved in PIRAD 5 versus PIRAD 4. And in fact, there are other people who have done work and showing that certain invisible cancers have got a genomic pathway. So they may be staying under the radar, stealth mode of a prostate cancer, which is using certain genes, which is not allowing us to pick up using our conventional imaging methods. But genomics can unmask them, and that all is happening right now. Then there are certain racial groups which are having more prostate cancer, and we have done an analysis as to what is going on, and certain factors are emerging which are immunological, which are biological, which are inflammatory, and we are finding certain PTEN losses. We talked about the PTEN as in one of the tumor suppressor genes. We talked about the epigenetic analysis that talks about the methylation, which may be happening more in African American. It helped me in making a better decision. It helped me in better understanding. It helped me in defining why a particular racial group is having more prostate cancer and which kind. But is it having an impact on me treating the prostate cancer? And answer is yes. Future is here because we have done couple of analyses in which we are looking at the immune checkpoint therapies and the targeted therapies. We can now know the patient has an androgen receptor kind of a problem or a neuroendocrine kind of a problem, which will have no impact from an androgen deprivation, rather will need a totally different kind of a treatment. We know which patient has an DNA repair pathway discussion, and so we can put them on the PARP inhibitors very early on, and who amongst these will be benefiting from the chemotherapy. All that can be done based on our understanding of this entire process. And also, we get to know not just the cancer cells, but the cells which are surrounding the cancer, what we call a tumor microenvironment. And tumor microenvironment is very rich with certain immune cells, and immune cells operate through those molecules, and these molecules are controlled by the genes. The proteins, molecules, genes, that interaction comes here, and we can be very precise in understanding how to boost up the body's own immunity to control this. And we have written these different papers. And one of them, which is we are very proud of, is we are doing what we call a personalized genomic vaccine. Cancer cell, in order for it to become cancer, it sometimes hacks certain normal control mechanisms. And in that process, it has some unique proteins. It brags about that it has those unique proteins, but that unique protein makes it a target because the normal cells in the body don't have that protein. Cancer cells, because of bad behavior, has developed those proteins or is using those proteins for their benefit. But we can recognize, we can train our own body's T cells and the NK cells to go and fight only those cells who have that abnormal protein architecture. We call it neoantigens. And we have a whole vaccine trial going on in which we are developing a personalized vaccine which will work for Mr. Smith or will work for another individual patient, and it will be very unique, very target, and it is happening as we talk. We have written books on this entire topic. I'm about to put together a second edition of this whole prostate cancer comprehensive book, and that passion about understanding the biology beyond just focusing on the nerve-sparing aspect, which is my full-time job. I really want to thank the organizer for allowing me to share my perspective about the genetics, genomics, and how it can have an impact in the patient care. Thank you very much.
Thank you. I think that was a very comprehensive and enriching talk on how cancer evolves into, and then specificities about prostate cancer. It is definitely exciting, although and there is some headway into clinics already, as Professor Ash mentioned. I think we can cover some of those issues during the panel discussion. So let's move on to our next talk. That would be by Professor Diane Thurusku. Thank you, sir. He would be speaking about genetics in bladder cancer, and again leading us on to a precision-based treatment option. Thank you.
Okay, well, good morning. Let me just start my PowerPoint. Okay. Good morning. And again, thank you very much for the kind invitation to speak today. What I'd like to do is, in the next 20 minutes or so, give you a glimpse of really cutting edge and very recent data that is really potentially both enhancing the precision and also the therapeutic impact and advances, leading to advances in bladder cancer. And I'd like to thank, again, the organizers for inviting me here to talk. So these are my disclosures. So for this audience, I don't think this slide is very new or surprising. Bladder cancer is a very important disease. And in bladder cancer, we are fortunate that most patients present with non-muscle invasive disease and have an excellent 5-year survival. However, about a third of those patients, patients with bladder cancer, present with invasive disease, which is invading into the muscle wall. And unfortunately, despite aggressive treatment, mostly with cystectomy, and in some cases radiation, their 5-year survival is about 50%. So in aggregate, most deaths from bladder cancer are due to advanced and metastatic bladder cancer. So over the last 20 or 30 years, a number of therapies have evolved to treat advanced bladder cancer. Obviously, I just mentioned, let me just turn on the pointer here. I mentioned radical cystectomy. for the primary lesion. When the cancer has metastasized, we have, for the last 30 years, cisplatin-based chemotherapy, as could be seen in this Kaplan-Meier curve. You could also see in this Kaplan-Meier curve that the results long-term, or cures, if you will, long-term, are in the order of 20%. Over the last 5 to 7 years, immune checkpoint inhibitors, either with anti-PD-L1 or PD-1 inhibitors, have made a significant impact in patients with metastatic bladder cancer, either in the first-line treatment or in failure after chemotherapy. So, the issues that I'm going to discuss today are basically how do we improve precision medicine in this setting. So I put the small arrows here for precision to indicate the people in the Kaplan-Meier curves that actually get cured, if you will, with the current therapies that we have today. So these are the patients, 20% from chemotherapy, here about 30 to 40% with immunotherapy, and of course with radical prostatectomy, 50% if they're earlier. So these are the patients that get cured. Now, the real advantage here is to figure out which are the patients that get cured with a specific modality, and then use that modality on them. That is the essence of precision cancer medicine. So I'm going to talk about that and some recent advances we have made that have been published in very high-impact journals. So we're going to do that, and I'm going to start with that, and then I'm going to shift gears and show how we could take the patients that now do not do well with what is available, and how we have used molecular technologies in my laboratory to really understand why they don't do well and make them a lot better by adding various other inhibitors. So that is the therapy part of precision therapy, how to enhance and improve both precision and the actual effectiveness of the therapy. So let's start with precision. For many years now, we as a field in urologic oncology and general urinary oncology and basic science have really searched, much like in breast cancer, to really identify the subtypes of bladder cancer. And with the hope that by identifying the subtypes within either the non-invasive and the muscle-invasive bladder cancer, that we would be able to identify various prognostic groups into these, and these subtypes would then help us guide patients toward one therapy or the other, or at least give them prognosis. So this is a somewhat complicated slide showing the various subtypes for non-muscle invasive disease or muscle invasive disease. You may have heard of some of these. Now, the problem and the challenge with these subtypes is highlighted in this slide. And several groups have analyzed the various subtype systems developed by a variety of institutions. And you can see here, just the details don't matter except to say that there is kind of general agreement between the subtype classifications, which are done based on RNA genetic profiles. But we would also agree that it's not perfect. So clearly, there is a less than perfect agreement among the subtype classifications. But what is actually even more challenging is the fact that when you classify patients that have had radical prostatectomy with these subtypes, the Kaplan-Meier curves here separate kind of so-so. They're not great. The separation is not great. In the end, at 36 months, 48 months, the separation is not great. The other somewhat troubling thing is that when you treat these patients with chemotherapy, the subtype does not appear to remain stable. So if you start with these subtypes basal, claudin, luminal infiltrated. And then you treat them and you analyze the subtype that is in the cancer that has survived chemotherapy. Then you could see that there's a very much a difference between, in this case, the luminal subtype and what the subtypes are within the cancers that have survived chemotherapy. So that tells you really that we have a much more sophisticated and deeper issue here that we really don't understand in the cancer. So I think the reason for that goes back all the way to the 80s and the 90s when I was actually in grad school. And a very brilliant scientist called Gloria Hepner postulated really the concept, which has been born to be true, that tumors are heterogeneous. So what the subtype classification that takes all the tumor, grinds it up, does gene expression, RNA expression on it, shows the tumor looks like this. But in fact, the tumor looks like a bunch of different, say, cancer cells, all with different genetic machinery. So yes, they are basically all, if you will, all people, but they're all different. You know, some are better at tennis, some are better at hockey, some are better at math, some are better at writing poetry. You know, they're all different. They all look like people, but they're all different. This is what Gloria Hepner put together, and this is what is called tumor heterogeneity. The fundamental reason why cancer is difficult to treat is on the slide. It's tumor heterogeneity plus tumor evolution. Charles Darwin was right on so many levels, including cancer. He didn't know he was right on cancer. But the principles of the evolution of the species that he wrote really applies to cancer. And I'm going to go through this slide. So tumors start being heterogeneous. They're all cancer cells, but they all have little different aspects. So when you put selective pressure on them, either not enough nutrients or the ability to not have enough oxygen, basically the fittest, it's the survival of the fittest, like Darwin said, for the animals. And the fittest cancer cells survive and they overgrow the cancer. Then cancer wants to grow even more, so they go to other parts of the body, but not all cells can do that. See, that's why purple and orange, blue, yellow, and green deliver. And then when you challenge them with therapeutics, basically you kill off certain ones that are sensitive, but the ones that are not sensitive grow up and survive and kill the patient. This is exactly why cancer is difficult to treat. We have different populations and they respond differently to treatment. So we need to really get a grip on this biology. Until recently, that was not possible. So we had a hypothesis that, in fact, these therapeutic issues can be understood better if we understand tumor heterogeneity better. And if we understand the various heterogeneous cells and what drives them, we could potentially treat various populations that will come up during therapy and metastasis. We would actually treat them when the tumors are primary. So the metastasis and therapeutic resistance would never happen because we would kill them right when they're very early. That's the dream, okay? So this is the first slide of the realization of that dream. So we took 25 bladder cancers that are muscle invasive. So these are the ones that half of them kill patients. And we did a revolutionary new technique called single cell sequencing. So imagine that we take these billions of cells and we actually take one cell at a time and do gene expression analysis. So we look at thousands of genes within each and every single cell, and we do it over thousands of cells in this cancer. So we're going to actually look at not only the cancer, but also the normal cells that compose a tumor, because a tumor is a ball of cancer cells, blood vessels, and other things. So when you do all that, and you put it through a computer, the computers are able to do essentially what some of you may know as it's a much more advanced form of principal component analysis, and is able to use the genetic information to actually group cells. And when the computers do that, they basically show you a picture that is really pretty because it assigns different colors, and it shows you what the cellular composition is of the tumor. What we have discovered in this, I love it, it's almost like a painting, you know, in this painting of the cancer is we found what would be expected. We found immune cells, endothelial cell, fibroblasts, and importantly, subtypes of bladder cancer, which were known before. But these are pure now because unlike before, where if a tumor was going to be classified as, in this case, it would be called maybe yellow or maybe blue, one of the two dominant forms, but it would be a mix. Now we know that there are components. But what the important thing is, we actually found a new subtype of bladder cancer that has never been found before, which is the blue ball here. We called it CDH12 because that was one of the genes that was most prominent in the subtype. And in the rest of the panels here, we basically show some of the gene expression profiles that are in these various clusters. So these are the higher genes that are in the various clusters. So these are not all the genes, this is just representative genes, but in essence, what they tell you is the cluster of genes that are representing by the blue cells here, which is the new population, are this block here, and there are about 200 of them. And similarly, for the various others, there are different genes representing. This basically tells you that there are signatures for all these subpopulations that are specific. So when you look at this picture here, these are only the cancers, and what the cancers do is basically show you that when you classify them using the conventional classification that I showed you in the second slide, these populations are all over. So this really tells you why when you do subtype classification, they're not predictive, because the subtypes are basically mixing up all these cells without the precision of single cell genomics. So what is the importance of this? Well, it turns out that when we look at the gene expression of the various populations, only the new population is prognostic after radical cystectomy. The other interesting thing is that when you overlay various other gene signatures on this principal component map that I showed you earlier, there are very strong signals indicating stem cells in this blue population. So if you remember here, the blue ball is at the top. So imagine this picture here, and you see how it's red here. It's red for both teratoscore, pluritest, and also neuroendocrine. So these blue new population cells have very high signals for stem cells, neuroendocrine cells, and pluripotent cells, which is stem cells. The other thing that is interesting about them showing in this panel here is the fact that these new cells, CDH12, also confirm that they are strong in the neuroendocrine subtype using the classical subtype classification. So they're stem-like, and they have elements of neuroendocrine populations, which some of you know, the medical oncologists in the group especially, that neuroendocrine tumors can arise anywhere in the body. So these cells have the elements of neuroendocrine biology. The other interesting thing that we found was when we looked at these populations in response to chemotherapy, we found that these populations go up. So these are tumors that have low levels of CDH12 population, and with chemo they go up. If they already have high levels, they stay the same. None of the other populations go up with chemotherapy. So they're bad with prognostic with radical cystectomy, and they also appear to be going up with chemotherapy, suggesting that they are contributing to resistance. Finally, in terms of prognostication, what we are very, very shocked to see and pleased to see, of course, is that they are the best predictor for outcome for checkpoint inhibitors. This is data from the IMvigor 210, which is at a PD-L1 inhibitor and PD-1 inhibitor, and we see here very well stratified patients in response to this therapeutic by CDH12 and other prognostic factors here, such as CD8, PD-L1, and PD-L2. So this is outcome and survival is basically confirmed by response. This is the responders based on CDH 12 population. The more population you have, the more they respond. The less population you have, the less they respond. The other interesting thing is that compared to all other biomarkers of response, the CDH population component is the strongest predictor. So this is currently the best predictor of immunotherapy response that we have found compared to the literature. So the other interesting thing was that this population tracks very strongly with fibroblasts. Fibroblasts have been known to be an aggressive, a contributor to aggressive bladder cancer for many years, and this explains why, and it also explains how, using this computational analysis, the cancer cells talk to the fibroblasts via secreted factors. I don't have time to go into this in detail, but there's a lot of talk between the fibroblasts and the cancer cells. This, again, is a slide showing that the fibroblasts are very important in the population, and that the cancer drives the fibroblasts to increase in muscle-invasive disease, and there's specific forms of fibroblasts called FAP fibroblasts that are driving the biology. So again, in summary, it is clear that we've discovered a new subtype of bladder cancer, that bladder cancer is heterogeneous, that this new bladder subtype causes aggressive cancer behavior resistant to chemotherapy, but paradoxically, they're very sensitive to immunotherapy, and does this kind of work through stimulating fibroblasts to help it be aggressive and also possibly be immunoresponsive. So now we could use this knowledge to do an algorithm of how to treat patients based on the C3 score, which is the CDH12 population score. We call it C3 for several other reasons. And this C3 score could be used to decide on neoadjuvant chemotherapy or immunotherapy here. So in essence, this is, again, hypothetical because it needs to be tested in clinical trials, but it lays the intellectual foundation of how to treat. So basically what this could be used for is to figure out how to take patients that respond and make sure that you only treat them and not the others. Now, what do you do with the patients that don't respond? So improved precision does not mean improved cures. It means basically just treating patients that we know are going to respond with just those patients and taking the others that we know don't respond and putting them on clinical trials. But how do we make those clinical trials better? So here is the slide coming back to show you what we did to make the red arrows actually shrink. So basically cure more patients with improved therapy. So for this discussion, we've worked in several areas in my lab, but for this discussion, I'm going to focus on checkpoint inhibitors. So people are testing a lot of checkpoint inhibitors and a lot of combinations with checkpoint inhibitors because combination therapy is really the key to curing cancer. There are no cancers that I know of, maybe with one exception, that is cured with a single drug. So we need to do combinations, but as this very simple math calculation shows you here, it is not possible to try every small molecule with every checkpoint inhibitor in every cancer in every stage. Not possible. Too many trials. That would require too many combinations and too many trials. So what do you do? What do you do it to do it more elegantly and more mechanistically with a lot more insight? So that's me thinking. Okay, so the way we came up with a very revolutionary, if you will, approach. And the approach was to basically make a hypothesis, which is on this cartoon. So the hypothesis was that when you treat a cell with a checkpoint inhibitor, some of them will die, but the ones that don't die because there are other pathways that keep it alive. In this case, cartooned pathway A or pathway B, okay? So if you take the cartoon a step further, if you inhibit the checkpoint inhibitor, the cells are fine, but because they're kept alive by pathway A. If you inhibit pathway A, if you knew what it was, the cells are still alive because they need the checkpoint inhibitor to work. They keep the checkpoint inhibitor pathway targeted by the checkpoint inhibitor. The pathway targeted by the checkpoint inhibitor has kept them alive. If you inhibit both, the cell dies. Okay? So what we did is we did a concept. So we did a proof of principle experiment. So the proof of principle was, let's see if we take all the drugs that are being used in clinical trials now for bladder cancer and figure out what their targets are for the targeted agents and come up with a list, a small list, because this was a proof of principle to demonstrate feasibility of this concept. That was pretty revolutionary. And so may have been wrong. Take 34 genes out of these targets of drugs, and then we made an shRNA library, which is a library of genes that basically built into viruses. And the viruses infect cancer cells, and these cancer cells then are a population where the genes are knocked out. So these millions of cancer cells in the dish, many of them have different genes knocked out. So it's a library of cells with a whole bunch of genes knocked out. And we used a cancer cell called NA13, which basically we developed in our lab. And this cell line basically is somewhat responsive to checkpoint inhibitors, PD-1 or PD-L1, but doesn't cure the mice. So it's a good line to use for these experiments. So what we did is we put viruses in these lines, in these cells, then injected the cells, and then randomized the mice to checkpoint inhibitor or nothing, IgG control. And this is what the tumors are cartooned to look like. So in the IgG treated cells, you can see they're multicolored. Okay, so each color represents a different gene being knocked out, but all the cells are happy and growing. On the PD-1, you see some of the colors are gone. And that's because the cells that are missing specific genes are killed by the PD-1 inhibitor, because they're the cells that have the two pathways, and we figured out one of the pathways. So those two cells are missing two pathways, and they die. Fantastic, right? So this was, of course, a hypothesis. So then what we do is we take these tumors out and sequence them and figure out which cells are gone and which cells are not, and what genes track with those cells. So when you do that, we came up with a phenomenal graph, which is here. Turns out that without going into a lot of details, these are the genes that are in the cells ranked. One gene stood out, well, actually a few at the front here, but the one that we picked on was DDR2. This gene was missing in almost all the cancer cells that came out of this experiment when they were treated with checkpoint inhibitors, but was there when not. So that was a really fantastic finding, especially when we looked at human tumors here. These are expression panels of all these genes that came out in the model, and we found out that DDR2 was showing up as overexpressed in a number of bladder cancers. So not only was it found in our experimental model, but it was also relevant in human cancer. So what is DDR2? DDR2 is is part of a two-gene family of DDR1 and DDR2. They're really interesting because they're basically collagen receptors. And collagen is one of the most important proteins that holds us all together, the glue that holds our cells together. And it's a glue that also is made a lot by fibroblasts. So if you think back to the first part of my talk. Fibroblasts are really an important part of the cancer. Well, it turns out that they make a lot of collagen, and collagen stimulates them and also stimulates cancer cells that have DDR2. And turns out that fibroblasts also have DDR2. So this is a really interesting story. So the other interesting reason why we're captivated by DDR2 was because they're drugs that inhibit it. Not these drugs like dasatinib were not developed to inhibit DDR2. They're developed to inhibit other targets for Philadelphia chromosome positive leukemia, BCR-ABL, but they do inhibit DDR2, so they're useful for our purposes. When we look at DDR2 in bladder cancer, as I alluded to before, it tracks with advanced stage, muscle invasion, higher in multiple data sets, and also higher levels are bad prognostically in multiple data sets. So it's a very bad actor, okay, in terms of expression. So here is really the exciting part experimentally, therapeutically. If you knock down DDR2 in bladder cancer and DNA 13 cells, and then you treat the tumors with PD-1, you see here the various growth curves. And if you inhibit DDR2 alone, you don't do much to the cancer in this model. But look what happens in the orange line when you combine both. I mean, the cancers are basically cured. So we're pretty excited about that, but we want to extend it to other models and see what happens. And you see here in melanoma, these lungs, this is metastasis. These lungs are a lot pinker, normal, than the lungs with the other treatments. And similarly for breast cancer in this waterfall plot, the combination works a lot better, and each gene itself each intervention does not really work. So this is true, remarkable synergism of two agents. So now we moved on to the therapeutics that we all want as physicians. Let's see what happens with dasatinib. So this is in colon cancer. We extended to even different models. So this is dasatinib alone. This is no treatment, dasatinib alone. This is a checkpoint. Look what happens when you do the combination. We cure all the colon cancers. This is single mouse experiments and data. Again, you can see every line is a single mouse and a tumor combination. So sarcoma, similar result. So this was very, very exciting. And in conclusion, and we're doing this on a much broader scale now, we have now a tool that can predict how the combinations with immunotherapy can be done in a rational way without guessing. We know now by testing genetically, dozens and now hundreds and now thousands of different drugs by virtue of looking at their targets, what works best with immunotherapy. So we're doing this in prostate, we're doing this in glioblastoma and pancreas, 3 cancers that are very refractory to checkpoint inhibitors. And I want to show you a few more slides to tell you actually potentially that this is actually working. This is a virtual experiment. It's not my trial, but I'm reading between the lines. This is a trial that was done by Mirati Pharmaceuticals. I have no financial interest in them in any way, except to show you this. This is a drug, sitravatinib, which is being combined in bladder cancer with checkpoint inhibitors, nivolumab. And I think you could see from this waterfall plot, the responses are spectacular. Well, it turns out that they didn't know this. I found this after I saw this trial and I looked more at the drug. Turns out that sitravatinib is probably the most important inhibitor of DDR1 and DDR2 known. It's more potent than dasatinib. I didn't know this when I did my paper, but it turns out sitravatinib is even more potent than dasatinib. My suspicion is that the reason why we're getting such spectacular results is because they're essentially mirroring our animal experiments, where you saw the combination of DDR inhibition and checkpoint inhibitor cures almost all the animals. That's probably what's happening here. So in summary then, here's the dream. Precision using C3, CDH12, the special population that we identified offers more precision. So with tools that we already have, either chemotherapy or checkpoint inhibitors, we can identify patients that are going to be complete responders, near complete responders. They're going to look like that in their Kaplan-Meier curve. However, for patients that are not responders, we're going to combine them with rational combinations, such as DDR2 inhibitors, and their Kaplan-Meier curve is also going to look like that because we're going to make them better. They're not going to be dying from only checkpoint inhibitors alone treatment because we're going to combine them with DDR2 inhibitors or something similar that is going to make their therapy better. So with that, I'd like to thank you. Again, thank you for the invitation. And be happy to answer questions or do the panel or whatever you want me to do next. Thank you.
Thank you. Thank you, Professor Dan. Again, that was a very exciting landscape we are having for bladder cancer. And thank you for sharing some of your experiments, which I guess are going to be much useful in future. So I think we would take some of the questions. I don't see any questions in the chat box from the audience. So we would take some of the clinically relevant points as we do the panel discussion. Now we move on to our third talk, that is by Dr. Anurag Mehta. I've already introduced him. So he would be speaking about relevant cancer genetics and kidney cancer. Dr. Mehta, please.
So is my screen visible there? Is the screen visible?
Yes, sir.
Okay, so good evening all. And thank you, Professor Ravi Mohan and Professor Raghunath for inviting me. I will speak on the subject of renal cancer genetics, which is basically a work in progress. I will not go into the molecular oncogenesis, but I will actually speak on the potentially useful and robust marker that have emerged in the recent times and can be used in clinical practice. So let's take a peek at what are the current happenings. So we know that renal cell carcinoma is a very heterogeneous disease. It has diverse histologies. There are 14 types, 4 subtypes, and there are 8 entities waiting on the sideline to make it to the classification table. And these evolving complexities, lack of resources, lack of expertise and exposure makes about 10 to 25% of these cases not correctly identified. But the tumor typing has prognostic significance, and it also has a utility in selection of therapy, and can also help in sifting out those cases which are likely inherited. And around 5% or more than 5% of RCCs are inherited RCCs. So when we look at this table, we see that the bulk of RCCs are clear cell renal cell carcinoma, about 70%, and the 30% are non-clear cell renal cell carcinomas. And these are identified by either by their architecture or cell type or the site of origin in the kidney. And lately, there are subtypes which are actually identified by their genetic signature. So we have a Barth-Hogg-Dubé syndrome, STH deficient RCCs, fumarate hydratase deficient RCCs, MITF translocated RCCs, TCB1 mutated RCCs, and Alperia RCCs. And unfortunately, all of these, which are defined by their genetic signature, do not have unique histologies. And therefore, it becomes necessary that these biomarkers are available for testing to make the correct diagnosis. In addition, we have these groups of inherited cancer syndrome within the clear cell renal cell carcinoma, the von Hippel-induced syndrome, within type 1 papillary carcinoma, the hereditary papillary carcinomas, and these 3 in the red boxes, and a few more which need to be identified so that the patient can get appropriate treatment, and the testing can be expanded to kindred to find out the silent mutation carriers. So this is what actually is necessary for making a correct diagnosis and finding the inherited cancer syndromes in RCC. And the RCC is also very diverse in terms of the oncogenic drivers. Dr. Tiwari had talked of oncogenic drivers. There are 17 oncogenic drivers which have been identified till date. And these different oncogenic drivers bestow upon these tumors different attributes. which determine the prognosis and can sometimes be used as a deciding factor in the management, especially these four which have been starred here. And these cancers also have different oncogenic addictions. They sometimes are addicted to hypervascular state. They may be addicted to metabolic reprogramming, or they may have immunogenic inhibitions. These have already partly been used to create targeted therapies, but we don't really use biomarker to decide what targeted therapy is required. So in the face of this extreme diversity of RCC, we have three challenges. Can we help establish more precise diagnosis than available? Can further prognostication be done beyond the clinical and laboratory risk models? And can we use biomarkers to select an appropriate therapy? So let's look at some examples of role of biomarkers in improving diagnosis. So here is one case of a renal cell carcinoma, a large tumor. On morphology, looks papillary. It has a high-grade nuclear feature. And obviously, this should have been reported as a papillary renal cell carcinoma type 2. But fortunately, we could do this genomic IHC. We have this available. And we found that this tumor is deficient in fumarate hydratase. And it has accumulation of corresponding oncometabolite, which is 2-succinylcysteine. And this means that this cancer is for fumarate hydratase deficient RCC. This necessitates germline testing. And if this index case is positive for fumarate hydratase deficiency, then the testing should be spread to the kindred. Now, why is it important to identify the fumarate hydratase? Because there is no role of observation. Unlike other inherited cancer where you can wait till the cancer has become 3 centimeters in size, here in fumarate hydratase deficient RCCs, waiting is not permitted because of the early metastasis to bones. And it has been also identified that advanced fumarate hydratase deficient RCCs may show better response to combination of bevacizumab and erlotinib, which is otherwise not a standard of care in RCC. And likewise, we can do genomic IHC for succinate dehydrogenase B, and we can identify the cases which have inherited deficiency of succinate dehydrogenase. We can also do a genomic IHC for TFE3 and TFE-B, and if it is positive, one must confirm by break-apart FISH whether these are MITF translocated or not. And if this is necessary that we must do FISH, because if the FISH is negative, then many of these cases or some of these cases with TFE3 or TFE-B expression may actually show rearrangement of the ALK. And this is a newly defined category of renal cell carcinoma, ALK rearranged carcinomas. It's very difficult to classify. It is very aggressive, usually have sarcomatoid, rhabdoid morphology. And the only clue could be presence of some of these signet rings. And if you are unable to classify a tumor, you are saying presence of these signet ring. These may be the cases to do an IHC for overexpression of ALK or to look for break-apart FISH to see whether the ALK is rearranged or not. And there are anecdotal reports which say that these tumors, which are usually advanced tumors, can show prolonged and profound response to ALK inhibitors. And then we come to papillary renal cell carcinomas. Typically, they are classified into type 1 and type 2, but any uropathologist will tell you that about 40% don't fit neatly into type 1 or type 2. And now more and more often, we're classifying them as papillary renal cell carcinoma, not otherwise specified. And there is an emerging entity which is called Oncocetic papillary renal cell carcinoma. And all of these have very different survival statistics. The Oncocetic papillary renal cell carcinoma virtually never kills. The type 1 has a very good survival, but type 2 has the poorest survival, with PRCC, NOS having an intermediate survival, and it may be actually useful to separate them. And based upon the gene expression profiling, it was found that there are 3 proteins which are differentially expressed, the ATP binding cassette C2, GATA3, and carbonic anhydrase 9. And using these 3 IHCs, the laboratory can separate these 4 categories, and which can then be better aligned with the prognosis. So the message as far as the use of biomarker in diagnosis is that they can help establish and refine diagnosis in up to 10% of the RCCs. They can help identify inherited RCCs, which can then be confirmed by appropriate germline testing, and can also help in finding out the silent mutation carriers in the family members, who can then be given who can then be advised on risk reduction strategies. And the message is in papillary renal cell carcinoma that they can be better diagnosed into prognostically significant category using a combination of ATP binding cassette ATP binding cassette C2 protein, carbonic anhydrase 9, and GATA3. Now coming to the putative roles of biomarker in prognosis, and I'll actually speak on clear cell and papillary renal cell carcinoma only. And we all know that the hallmark of clear cell renal cell carcinoma is inactivation of a tumor suppressor gene called VHL gene. The VHL gene is inactivated in about 90% of the cases. In 67% of the cases, there is a biallelic inactivation, while there is a monoallelic inactivation in 22% of the cases, and about 10 or 11% of the cases have a unmutated or a wild type of VHL gene. And this knowledge, whether it is monoallelic, biallelic, or wild type, is very significant prognostically. It has been shown that those tumors which have no head, which are wild type or have one head, they have aggressive phenotype, they're sarcomatoid or rhabdoid, they have high ISEP nuclear grade, they show inflamed stroma, they metastasize to the regional lymph nodes early and also show distant metastasis early, and they have very heavy expression of VEGF. And as far as the cancer-specific survival is concerned, it's shown that those which are wild type have a median CSS of 33 months against those who have biallelic inactivation, where the median CSS is about 107 months. So this is a very significant finding. And I think in the new WHO classification, probably VHL testing will become mandatory to diagnose clear cell renal cell carcinomas. Sorry. But VHL inactivation by itself is not adequate, and over a period of time, these tumors acquire additional genetic alterations, and these genetic alterations are largely in chromatin remodeling genes, which Dr. Ash Tiwari had so eloquently spoken about. And there are 4 of them: polybromo 1, lysine demethylase 5C, BRCA-associated protein 1, and SADG2. And in addition, there are about 5% mutation each in the genes of the PIK3CA-AKT-mTOR pathway, the TERT promoter mutations, and loss of 9P and 14Q, and about TP53 in 2% of the cases. So why is this knowledge important? It has been seen that this is prognostically very crucial. So VHL, we have already spoken. So let's see how PBRM1, BAP1, and TP53 inactivations are associated with survival. And it has been shown that those tumors which have a PBRM1 loss of function have a far longer overall survival in this study, 10.6 years, against those who have BAP1 inactivation, which had a OS of 4.6 years. And the same finding was also seen in the TCGA database. And this is basically all stages and patients both on TKIs and in the pre-TKI era. And it has also been shown in this mycologist study that BAP1 inactivation versus those which are wild type have a very significant difference in survival. And it is seen that even when the S sign score is low, but the BAP1 is inactivated, the prognosis is very, very poor. So BAP1 inactivation is an independent marker of poor risk, independent of the S sign score, at least in the low S sign score category. And coming to the advanced and metastatic RCC, this MSKCC study has shown that tumors with TP53 inactivation and BAP1 inactivation have a inferior OS, while those with the PBRM1 inactivation have a better OS. And this they have tried to incorporate into their MSKCC score to improve its C index, but that has not been validated so far. And then we come to papillary renal cell carcinomas. And the papillary renal cell carcinoma, of course, as I said earlier, it's very significant, important to separate them clearly and correctly. And then the pangenomic data from the TCGA has actually separated out a cancer type, which is characterized by CpG island methylator phenotype. And in this category, there is a silencing of several genes by methylation of the promoter sites. And two significant ones are P16 and fumarate hydratase. And you really don't have to do the methylation studies. You can do the IHC, two IHCs, P16 and fumarate hydratase. If both are deficient, then this will identify this category, which has the most dismal prognosis of all renal cell carcinomas, including the clear cell renal cell carcinomas, the worst category, and can easily be identified by doing just two immunochemistries. And then to clearly separate out PRCC1 and PRCC2, the LET7 family of microRNA have been studied. It has been shown that PRCC1 has a varied expression of these microRNA, while PRCC2 shows universal expression, tenfold expression, or up to tenfold increased expression of this family of microRNA. And this can actually really clearly separate PRCC1 and PRCC2 in a significant, in clinically significant fashion, prognostically significant fashion. So the message here will be that in clear cell renal cell carcinoma, BAP1 inactivation is bad prognostically, both in localized and metastatic RCCs. TP53 inactivation is bad for metastatic disease, and PBRM1 inactivation has confers good prognosis in both early and advanced clear cell renal cell carcinomas. In papillary renal cell carcinomas, absence of immunostaining for P16 and fumarate hydratase identify a subsets of papillary renal cell carcinomas with CIMP, CpG island methylator phenotype, which has an appalling prognosis. An expression of LET family of microRNA can separate PRCC1 from PRCC2. And then let's come to the predictive role of biomarkers in metastatic clear cell renal cell carcinomas. And what we use here are TKIs and IO combinations, or two IO combinations, or we use TKIs alone. So are there any predictive marker for benefit from either one of these or either of these? So let's look at the predictive markers for tyrosine kinase inhibitors. And in this Dana-Farber study, where the two polar groups were used of extreme responders and those who had a refractory disease, they didn't respond to sunitinib at all. And when their whole exome sequencing data was examined, it was found that the extreme good responders had significantly increased inactivation of PBRM1. So PBRM1 was the only gene which was significantly more inactivated in the extreme responder compared to those who were refractory. And the value of PBRM1 inactivation has further been demonstrated in iMotion 150, where it is shown that PBRM1 inactivated tumor respond better to sunitinib with a PFS advantage of about 15 months, compared to when they are treated with the atezo plus BAB. And the same finding has been reiterated in record 3 and COMPARS. And an interesting finding which has been shown in the record 3 cohort is that those tumors which have inactivation of lysine demethylase 5C, when they are treated with sunitinib, they have an excellent median progression-free survival, 20 months against 8.4 months when the KDM5C was wild type. And it has also been seen that patients who have high angiogenic signatures respond better to sunitinib, and this has been confirmed in iMotion 151. And when we come to the use of ICI in advanced clear cell renal cell carcinomas, none of the conventional biomarker for immune checkpoint inhibitor really works in RCC. There is no you can't use tumor mutation burden, no association, neoantigens, tumor indel burden loads, or inflamed phenotypes. So is there anything else that can be used to predict response to immune checkpoint inhibitors? And again, the PBRM1 stands out. It has been shown that those with PBRM1 inactivation show more clinical benefit and a somewhat better PFS and OS compared to when they are pitted against mTOR inhibition in this group. And in this study, it has been shown that tumors which had a PBRM1 inactivation had many more than those which were wild type had objective responses. This dendrogram shows many blues here, means these are PBRM1 inactivated which had higher objective responses. And it has been seen that the RCCs which are inflamed, which have a high CD8 positive T cell infiltration, don't show any clinical benefit with anti-PD-1 treatment, no benefit in PFS, and no benefit in OS. And this is because most of these cases have deletion of 9p21.3, and this deletion is mutually exclusive with PBRM inactivation. So the tumors which are T cell effector, T cell infiltrated and have this del 9p21.3 loss, these are the ones who don't respond to immune checkpoint inhibitors. And in iMotion 151, it has been shown that tumors with high T effector signatures and low angiogenic signatures are the ones which can respond to atezo plus PEP combination, completely opposite to the TKIs where high angiogenic signature and other low signatures were associated with better PFS and OS. So are there any predictive marker for papillary renal cell carcinomas? And the type 1 papillary renal cell carcinomas, about 40% have MET alterations. And one will expect that MET inhibition will be a natural choice of a drug for papillary renal cell carcinomas. However, so far no success has been achieved with the MET inhibitors. Probably, there are phase 1 and phase 2 trials now with the MET-specific inhibitors, capmatinib and tepotinib, and probably they will give us better result for MET inhibition in papillary renal cell carcinoma. So, to conclude the third part of my talk, PBRM1 inactivation in clear cell renal cell carcinoma predicts response to VEGFR TKIs and IOs, though there is no approval of immune checkpoint inhibitor monotherapy in first line. It is inversely related to presence of T effector cells and del 9p23. IC immune checkpoint blockade plus VEGF antibody use is beneficial in patients who have high T effector cell signatures and low angiogenesis signatures. Lysine demethylase 5C is a strong predictive marker for prolonged benefit from sunitinib. Despite MET alteration being common, we haven't found this to be actionable so far, unlike in NSCLC. And my request may be, we can all start doing at least testing for few of these genes, VHL, PBRM1, BAP1, SATD2, KDM5C, and TP53, to help in prospective validation, in prognostication, as well as in therapy selection. And it must be said that this is a work in progress because none of the biomarker has prospective validation to recommend its use. So with that, I will close. Thank you so much for your patient listening. And thank you, the organizers, for inviting me.
Thank you very much, sir.
Yeah, I request Dr. Sanjay to...
Raghu, we can't hear you.
Hello, Sanjay, can you hear me?
Yeah, okay. Yeah.
Sanjay?
Yeah, I can hear you.
Yeah, thank you, sir. I request Sanjay to go ahead with the panel discussion. We have shortage of time. I request Sanjay to manage that. Thank you.
Just looking at the time, it's 8:26. So we'll try to finish by around 8:45. So 20 minutes. Thank you very much for this opportunity, Sogo, Mr. Raghunath, and Dr. Ravi Mohan. I have got this panel, but I think Mohan is having problem with his connection. So I'll try to use Ginil, and Professor Ganesh Bakshi from Tata Memorial is also there. So though he's not on the faculty, if there are any questions, I'll ask. And international faculty, just for me to spell your name right, sir, is it Dan Theodorescu? Hello?
Yes, I'm switching microphones.
Okay.
Yes, it is.
Can I just mention as Dr. Dan, sir? It's just...
Yes.
Oh, apologies for being disrespectful. And Professor Sujit Nair and Professor Dimple Chakrabarti.
Thank you. Yeah.
Okay, thank you. And it was so nice hearing your talk, sir. Dr. Dan, the reason is my MD was on isolation and characterization of stem cells. So to listen to that word stemness and isolation of stem cells, it was very good. I'm just going to start with this one. Okay, so this is about, I will say, worldwide variation in prostate cancer related mortality. So the red is mortality, as in high mortality, blue is less, and we happen to be half and half. Okay, so I'm just going to ask Dr. Sujit, Professor Sujit Nair, if you're around.
Yes, I'm around. Yes.
Do you think this mortality is down to race, genetics, or lifestyle, or all of them?
It's actually all of them.
Which one is the highest component causing this?
So it's actually a mix of everything. It's the genetics plays a very important role, as you might have heard from Dr. Tiwari's talk, that how the genetics and the whole makeup of the cancer, and from Dr. Dan's talk, you heard about the heterogeneity of the cancer. So it's the genetics, the heterogeneity, and it's further compounded by certain race and the lifestyle, which either in most cases a poor lifestyle and being of a certain race has more predisposition to the cancer and it's more aggressive behavior. So it's actually, you cannot actually say one is more Okay,
I'm going to request this one. If you were going to rank them as the most important, second most important, third most, what is it?
So I would say genetics plays an important role. Yes, that's first. And then you could say the race, and third, lifestyle.
Okay, in that order. So I'm just going to ask, is it genetics because of lifestyle? And Dr. Dancer, is prostate cancer preventable? And what would you rate them as? If prostate cancer, I know your specialization is with respect to the bladder cancer, but which one do you...
this is a very, very interesting. I love, and cancer. One of the things that really strikes me is the fact that if you look at latent or cancers, across the world, those rates have remained remarkably similar. And so what that tells me is that the underlying biology, although there are differences in certain areas, there's a huge environmental component. Because if you look at being the latent cancers the same, and yet the clinical cancers being different, especially if you look at migration studies from the Japan, for example, to Hawaii and to United States, then basically you see that there's a very strong environmental potentially in doing this. So I would say that the environment plays a very, very significant role, probably as much as the genetics.
So you would...
Now, when I say genetics, I mean, non-germline genetics. For example, there are clear things like, for example, in breast cancer, BRCA, right? BRCA1, BRCA2. It's a different animal.
Okay.
But I'm talking for non-major germline issues. I think environment is very strong in prostate.
Okay. And I'm going to ask the second question. Among the three cancers that we have had talks on, which one is most preventable? Dr. Dan.
Well, I could argue that both bladder and prostate would be potentially preventable. I think bladder we've tried to do prevention.
Okay.
There's really nothing yet short of, not smoking. That's the number one major cause, as we all know, bladder cancer. So that's preventable in that rate, not smoking. Number two, exposures to carcinogen. Facial things is now mostly gone. And I think the third thing is, and I work in this area, we published in this, is the androgen receptor.
Oh.
Be quite important in bladder cancer. We and others have shown. And although we don't really know exactly its role in advanced cancer, it seems to be working both as a promoter and potentially as a support.
Okay.
So I'm not going to go into too much detail, but what is, I think, fairly solid is early disease. In the trials that have used 5-alpha reductase inhibitors for BPH and androgen deprivation for prostate cancer, those studies have shown a remarkable decrease in bladder cancer. What that tells us is that bladder cancer potentially could be preventable by altering the androgen axis, which is kind of interesting, right? Now, that doesn't mean that it's going to be treatable with androgen deprivation if it's established. It's a different, I want to emphasize this very fundamental difference between prevention of bladder cancer and actual disease with androgen deprivation is a viable option, because there's, and I'm happy to extrapolate more if you want.
Just going to ask this question, sir. If there is that much dependence with respect to the androgen receptors, how do we explain the cancer in females? Do they have androgen receptors as well?
Oh, yeah. Well, great. Thank you for asking, because there's a lot of question of what about androgen receptors? Well, let me tell you this. A couple of observations. Androgen in breast cancer to be a very important driver of triple negative disease. Triple negative disease is a form of breast cancer. Androgen receptor, as a matter of fact, enzalutamide has been tested. So, take-home message. Androgen and androgen axis is important in women. That's a take-home pearl here from the meeting. So, therefore, we do not have a human experiment with 5-alpha reductase inhibitors in women, of course. But so we can't really show that that's relevant. But potentially discount the importance of the androgen axis in women just because they're women, as the breast cancer literature would suggest.
Thank you. Ravimo, is your connection fine for me to ask a question? In India, we are seeing an increasing incidence of prostate cancer, which is what I was going to show. That means that it seems to be related related to lifestyle. So, and you know what the other two people have answered, but do you think, especially with respect to prostate cancer, would you like to rate them as to what is your order of causality with respect to prostate cancer? Lifestyle.
Yeah, I think, am I audible?
Yes.
Yeah. I think I would perfectly agree with Professor Dan that it's the epigenetic mutations which are happening because of the lifestyle changes, and that is how. And as we are moving from what we were to what we are, that is from a developed to a developing to a developed world. I think that we are also having a lot of epigenetic mutations, and some of them are responsible for increased incidence of cancers in general. For us, it is more of prostate cancer, but in general, you see the cancers are on the rise in India.
Thank you. So I'm just going to talk about bladder cancer because that is what we were discussing. I'm just going to ask a comment first from Dr. Dan. Is this a right statement? And if you can be concise in saying that, why is it wrong? A patient started off, because commonly a junior doctor would present a case saying that a patient presented with a low-grade carcinoma, urothelial carcinoma in 2018, then had an aggressive non-muscle invasive bladder cancer, now has got a high-grade muscle invasive bladder cancer. Is that a wrong statement or right statement?
So I'm not quite sure what you mean.
What I mean to say is that kind of low-grade...
He has 3 lesions, right?
No. This was in 2018. He only had low-grade cancer, was under control, then had high-grade NMIBC, now has got high-grade muscle invasive bladder cancer. So in another way, what I'm trying to say is, can a low-grade cancer become a high-grade MIBC?
Well, actually, yeah. Okay, I understand what you mean. So, I think that that's probably not the same cancer. And the reason why I say this is because there is a very clear genetics that track with low-grade disease versus high-grade disease. And it is very unusual for those genetics to actually flip within a certain population. So I suspect that given what we all know, that bladder cancer is a multifocal field disease, it was a low-grade urothelial carcinoma, and then the recurrence, the second one, the aggressive NMIBC genetics, is from a different transformed focus, and it is that cancer that then progressed to high-grade MIBC. I think the last two are probably the same cancer, but the first one is probably a different cancer in the same damaged urothelium.
So you can see this kind of a presentation, but it isn't the same cancer that is evolving. It is a different cancer in a field change.
That would be my guess. And it's a very easy guess to prove, right? Because all I would need to do is to do a sequencing analysis on those 3 cancers, and I would know instantly.
Okay, I'm going to request Professor Dimple Chakravarti, if you are around. Is there anything called as low-grade MIBC? Genetically, is it possible? Or Sujit Nair?
Sorry, this is not my area, so I will defer that question to Dr. Dan, okay?
Is there anything called as a low-grade MIBC, sir? Dr. Dan?
No.
Okay. Low-grade... I just wanted an answer like that. That's fine. So, low-grade urothelial carcinoma with metastasis, yes or no?
No.
Okay, thank you. Then I'm going to ask Dr. Ravi Mohan, high-grade NMIBC successfully treated with induction BCG, just with induction BCG, at 3 months, now only has a low-grade non-muscle invasive bladder cancer. Can we say that it is successfully treated?
No, I would say, and I think these would be the patients where we actually need to do some genetic testing by next-gen sequencing to identify, because if that theory, what Dr. Dan has just mentioned, is if it is correct, that there is no flip between a low grade and a high grade, then the recurrence of a disease or finding a new disease, even if it is qualified as low grade by a pathologist, would not actually mean that the patient has been treated or cured. So I would, with that theory, but unless we have done good amount of genetic analysis and found out for ourselves, it's difficult to answer.
Dr. Bakshi, would you take this as a success story, or I don't?
The last one?
Yes, the last one.
See, the last one kind of loosely fits into one of the definitions of BCG failure. So it is a failure, of course, by definition.
Dr. Dan, any clarification on that?
Yeah, I like definition. They're good for clinical trials and really structuring clinical investigation, but I also like to think at the biological, more refined, sort of splitting hairs, if you will. So if you look at BCG and what it does, basically BCG in an ideal world prevents progression and prevents. Those are the two things, right? And you can't prevent recurring, but you can recur with, right? And this person basically has a recurrence that is non-progressive. So I think, again, I don't want to feel like I don't want you all to feel that I'm splitting hairs, but happened is that it seems that the high-grade component or therapy response, whereas the low-grade or perhaps a second cancer has arisen in this setting. So I suspect that if you did sequencing of the original high-grade MIB and the low-grade that's recurrent here, I suspect they're going to be different.
Thank you. So
one question, Dr. Sanjay.
Yeah.
Can we? Is it possible that BCG actually changes the genetic makeup of these cells? Maybe Professor Dan, if you want to take it.
Yeah, I'm happy to take it. So the way we have a paper that's in review now at a major journal about BCG resistance and success in the genetic. In the discussion, but I think what's happening is if you take the cancer, if you remember the slide that I showed you with the what's called the UMAP plot, which is that nice color picture of the principal component. So imagine the BCG sculpting that population. So it's a sculptor that basically says, oh, I liked all those colors, but I don't like blue. So I'm going to take that away. A picture that are going to be eliminated. It really change the genetics, but it actually changes that the cancer is composed of, the tumor is composed of. So in that way, it changes the overall genetics as a population, but the cell either gets destroyed or persists. Okay, it doesn't actually change the genes. It just, and then Darwinian selection basically knows that the BCG doesn't kill them indirectly through the immune system. So that's how I think about the problem.
I've got another 2 minutes to go, so I'm just going to. So this is, I know it's a very old paper, but this is how we used to. Actually, this isn't the question that I wanted. So the question that I wanted to ask was, this was the standard classification that we have been using for many years. Low grade, high grade, NMIBC and MIBC. And then now more and more, there is more scientific basis for this. And I'm thankful to Dr. Santosh Menon from Tata Memorial for sharing this. Are we going from physical identification, which is what we have been having for all these years, to something a bit more intelligent, bit more digitalization of everything, because these are all numbers. Whereas before, everything was physical. What you saw was what you got. And the last part of it, now we are seeing the results of this. Do you think this is going to be the future of how we are going to classify? Dr. Dan?
Well, as I said in my talk, I think the subtypes are okay, but there's a lot of issues with them. The agreement, there is consensus in the molecular classifications, but in fact, I think the era of taking bulk cancers and sequencing them either with RNA or DNA is pretty much, most of us that work in this field at the cutting edge feel that era is probably going to be over soon. And the reason is because as you've seen from my presentation, When you look at the actual single cells, it's very complicated inside the cancer. So when you take these subtypes, you realize that you're taking a cancer, you're grinding it up. So these subtypes are RNA based on RNA expression, everything in there. Cancer cells, normal cells, and a whole bunch of different cancer cells. So these will never be as precise. And so if you want to look at the tumor architecture, I think this is the past. The future is single cell technologies in blood, in tumors, in urine. We have a concept called the molecular twin, which is doing multi-omic analysis at the single cell level on the cancer using proteomics, DNA, RNA, and machine learning on the tissue slides, etc., to bring it together. So I think this is the past. The future is single cell analysis.
So, Dr. Sanjay, I would like to add something to this point here for the elegant description given by Dr. Dan, is that any classification you consider now is subject to change because of the technologies are emerging and they are evolving every day. And so just like what Dr. Dan was saying, here we have certain technology that allows us to measure single cells in the urine of prostate cancer patients. So these diagnostic tools and the classifications are going to keep changing, but even though the broad structure remains the same, but more granular information, more information at a very singular cell level will continue to emerge. So,
thank you very much. Sorry, I could not include everybody in the panel, and we could not go for as much time as we wanted, but we were already running late. I'm very grateful. It was very interesting talk, Dr. Dan and Dr. Ash Tiwari, sir. And I would hand it over to Ravi Mohan and S.K. Raghunath to conclude.
Yeah,
thank you.
Thank you, Dr. Sanjay. You made it interesting, even though for a very short time, and I apologize sincerely for that. There's one question from the audience from Dr. Sumit Day. He's the PhD with me. His question to Professor Dan is, what do you think about conditional reprogramming of patient-derived cancer cells in future as a part of precision medicine? Professor Dan?
Yeah, so thank you for that question. So when you say conditional reprogramming, I'm not, there's multiple ways to do that. Maybe you could tell me more what you mean.
So conditional, we derive the patient cancer cells, reprogram it so that those cells start responding to a particular therapy or start behaving, and then inject them back into the patient. Yeah. And see whether the tumor is. So he's actually working on a concept like that. He's a PhD with us, but his question is, how do you see it as a possibility?
Yeah, so I think it's the way thinking about some of these concepts, but more in the sense of we would like to take cells and reprogram them to actually express certain things that make them more immunogenic. And so that concept is old. I remember reading about that when I was in high school, about people trying to do cancer vaccines. We haven't published this yet, but we're very excited because we found a new way to do angiogenic that nobody else has identified before using a technology that I invented. And patients reprogram them to express these antigens that have not been previously thought to be vaccine antigens, and then put them back in. And I think that's very promising. Whether taking cells to differentiate them, do other things, but I think all the maneuvers to take cells to somehow, I think, trigger an immune response. Because otherwise, I guess beyond that, how do we do the reprogramming? What else do we do to reprogram them successfully to destroy the parental cancer?
Okay, thank you. Thank you very much. I think that answers a part of it. And Dr. Sujit has shared his trial, so maybe that's an interesting.
One of Dr. Tiwari's trial in personalized genome vaccine. So I thought.
Yeah. Perfect. Thank you. So over to Dr. Raghunath for his final comments for the program. He's the main person behind it.
Thank you, Dr. Ravi. Actually, Dr. Ginil is going to sum up briefly. We are running behind schedule. I request Ginil to sum up.
Thank you, Raghunath and Ravi Mohan, for giving me this important responsibility. We had 3 talks in this program. First was by Dr. Ash Tiwari. He explains the very basics of genetics in a way surgeons can understand. It's important to see that 71% of CA prostate has got some abnormality in androgen receptor pathway, 49% in PI3K pathway, and 13% on DNA repair. And this is going to be an important factor for risk stratification, prognostication, and planning the treatment. Professor Dan's the talk was very informative. He explained about genetics of bladder cancer in a clinician's perspective. He highlighted the fact that cancers are so heterogeneous and there is need for personalized treatment after properly subtyping the bladder and knowing the exact tumor biology. His research on single-cell sequencing of MIBC was very interesting. CDH13, that's C3 scores for prognostication and DDR2 inhibition in treatment, etc., may be the future of bladder tumor treatment. Dr. Anurag discussed about different pathological variants and where we have to go beyond the pathology, like genetic screening, and also explain different markers that can predict the tumor response in various treatment. My final comment is that tomorrow is going to be individualized, personalized care of cancer, and it will be like doing a culture and treating infection. We are going to be having a genetic sequencing and treating the patient according to the specific alteration that has happened. And for us as a surgeon, the time is changing, and we have to have a lab beside our OT and individualize the treatment. That is the way in which it is going to be. Thank you very much.
Thank you, Dr. Gill. With this, we have come to the end of the scientific program. I thank all the international speakers and the national faculty for their contribution. And I thank Dr. Ravi Mohan. Within just 7 to 10 days, he made up this program and conducted this program in an excellent manner. I thank Dr. Sanjay. Always, he is there to conduct our panel discussions in the best possible way. Thank you, Dr. Sanjay. I thank all the panelists. I thank SOGO office bearers and all the delegates. And at the last, I would like to thank Ferring for their support. Sorry for overshooting time. We took more than 25 minutes extra. Thank you all. Good night. Thank you.